Hello, my name is Estefania Torrejón, and I am going to present the work EVNet, Extracellular vesicles-mediated intercellular communication networks involved in metabolic disease pathophysiology. This work was done in my home institution, Nova Medical School from Lisbon, Portugal, and in cooperation with the Systems Biomedicine Lab from Anaïs Baudot in Marseille Medical Genetics. At the moment, there are many cell-to-cell communication tools, and most of these tools rely on RNA-seq transcriptomic data. They work by identifying ligands expressed in donor cell populations and receptors expressed in recipient cells, and then by cross-referencing these pairs with curated interaction databases. These tools can infer potential communication matches between cell types based on gene expression levels. Then we identified this tool called NicheNet that can not only identify these cell-to-cell communication matches, but can also predict the effect of these interactions in the recipient cell. In my lab, we study extracellular vesicles, also called EVs. They are bilayer nanoparticles that act as carriers of diverse biomolecules such as proteins, metabolites, and some RNA sequences. And they have an important role in intercellular communication and can be uptaken by the recipient cell through different mechanisms such as ligand binding, endocytosis, and membrane fusion. As we study EV's communication interactions in my lab, we decided to modify NicheNet to be able to study EV's mediated communication. NicheNet applies a personalized page-run algorithm on a prior knowledge network. This is an example of the network. And here the ligands of interest are used as a seed node to compute the probability of influencing downstream target genes through network interactions. However, extracellular vesicles engage with the recipient cells via additional mechanisms beyond ligand binding, like I mentioned. So in this context, we apply NicheNet's scoring algorithm, not only using the ligands as seed nodes, but also using downstream proteins and transcription factors. So we basically use all the nodes that you could see here as the seed nodes. So our tool, EVNet, enables the prediction of the effects of the EV's proteomic or transcriptomic cargo on recipient tissues of interest. So users can input differentially abundant proteins or differentially expressed genes in the EV cargo across two conditions, along with differentially expressed genes from the receiving tissue across two conditions or also two distinct cell populations. And so EVNet supports datasets from Homo sapiens and Mus musculus. Now as use case, we use data that were already published from my lab, in which we wanted to characterize the proteomic profile of gut-derived EVs isolated from two different groups of mice, one that was under a normal diet and the other one was under a high-fat diet that also induced pre-diabetes in the mice. In this group, we also saw, checking at the results from the liver histology, that they had a fatty liver disease phenotype, and essentially the proteomic profile between the cargo of these EVs was different across groups. Then we knew that these extracellular vesicles or EVs were going to go to the liver, but we wanted to see to which specific cell population in the liver they were gonna go. So we had another experiment in which these EVs were injected into a second cohort of mice, and we saw that over 65% of these gut-derived EVs were uptaken by Kupffer cells. So we wanted to use the developed tool EVNet to see the effect of this EV cargo on Kupffer cells. For this, we use differentially abundant EV cargo, in this case proteins, derived from gut-derived EVs isolated from mice under two conditions, the healthy and pre-diabetic. This was for the case of the EV cargo, and then for the recipients population, we use differentially expressed genes obtained from the tabula muris single-cell RNA-seq dataset from the tabula muris consortium, and we did differentially expression analysis between Kupffer cells and hepatocytes. Results. We are focusing on the differentially abundant proteins on the pre-diabetic phenotype for the sake of time. And we can see here the top 30 that have a high regulatory potential on these upregulated Kupffer cell genes. And it just comes to our attention immediately that we have this protein here with a high regulatory potential on many Kupffer cell genes that is called SCLE. And it goes, SCLE goes for selenocysteine lyase, and it's an enzyme involving the recycling of selenium from selenocysteine during degradation of selenoproteins. So it's involved in the selenocysteine biosynthesis and involved in redox balance and attenuation of Kupffer cells pro-inflammatory activity. Then we could also with EVNet do this interaction network in which we saw direct interaction with the Kupffer cell gene SEB2 that in Kupffer cells maintain cell identity and their survival, but also inhibits TGF-beta1 activity, that is a cytokine that is involved in the fibrogenesis and development of hepatic fibrosis. As a home message, we think that EVs play a crucial role as agents of interorgan communication and could be key contributors in the pathophysiology of fatty liver disease. Pre-diabetic gut-derived EVs carry SCLE to Kupffer cells, which play a role in redox balance and attenuation of pro-inflammatory activity. It also activates SEB2, linked to macrophage survival and inhibition of fibrogenesis in the liver. These interactions are EVNet predictions and will also get validated experimentally, and this is just a use case. EVNet can be used with other sets of EV cargo and in other recipient tissues of interest. Thank you very much.